Autonomous specification, as described in the slide, is a form of embryonic specification where a developing cell differentiates and turns into a specialized cell without any external signals. It is due to cytoplasmic factors or determinants which helps a cell commit to a certain cell fate. Autonomous specification results in mosaic development, that is, if you try to remove some specific cells of an organism, you will see that some part of the developed structure vanishes or does not develop completely as the cells responsible for that part have been removed and no other cells can replace its function. Autonomous specification is majorly seen in invertebrates. Cells undergoing autonomous specification know their fate very early on and show a fixed pattern of cell division. An example of autonomous specification can be seen in snail patella. The figure A in the slide shows a mass of cells, zygote, which is successively undergoing cell divisions to form more cells as can be seen in figures B and C. It was assumed that the pink mass of cells in the figure was trochoblast and that was isolated during an early stage as can be seen in figure D. It was found that even after isolation, those mass of cells formed a trochoblast. This suggests that the cells knew very early on that they had to form trochoblast and hence showed fixed cell division patterns and did not require any communication from neighboring cells. Another example of autonomous specification can be seen in tunicates. An experiment was performed and the scientists and embryologists had noticed that the yellow-colored cells that can be seen on the slides developed into muscle cells later on. In order to confirm this, they isolated the cells which they thought formed muscles. These cells were called B4.1 and they cultured them. It was found that those cells did in fact develop into muscle tissues. Then these cells were removed from some part of the early embryo of the tunicates and it was found that the tunicates did not develop to have tail muscles as it can be seen in the last figure. No other cells could replace the function of these B4.1 muscle cells and hence tail muscles were absent. This confirms autonomous specification.
The second type of specification is conditional specification, and this is when the fate of the cell is determined by its position and proximity to neighboring cells. This will dominate in vertebrates, but can be seen in some invertebrates, and this is what will ultimately lead to cell rearrangements and migrations. In this example of conditional specification, we see a tadpole blastula, and we can see the difference in development between back and belly tissue. So we see in a normal blastula that back cells will form back tissue, but we see when that back tissue is transported to belly tissue, it will ultimately form belly tissue due to what cells are in proximity to the tissue inserted.
Syncytial specification is the last of the three and it shares components of both autonomous and conditional specification. It can be considered a hybrid of the two, however, cell cleavage is variable, so there are no rigid cell fates for any of the particular nuclei. The fate of the cells is determined based on their positioning amongst the cytoplasmic nuclei, which develops a gradient. This gradient acts as a morphogenetic determinant. Most insect classes undergo this type of specification, and one of the best known examples is the process that takes place in the fly Drosophila melanogaster. As mentioned previously, one of the best known examples of syncytial specification is the process in Drosophila melanogaster. Figure A shows the progression of the cellularization of the blastoderm. The developing blastoderm undergoes cycles to produce nuclei, which originally stay in the middle of the structure. During cycles 8 and 9, the nuclei start to migrate towards the outside cortex while some remain in the yolky region. Pole cells then begin to attach to the end of the developing blastoderm. In the later cycles, cell membranes begin to form between the nuclei and cellularization of the blastoderm occurs. In Figure B, you can observe the division of the nuclei that takes place during cycle 13, and you can start to see asynchrony in the cell division. This figure gives a visual to the gradient created by the positioning of nuclei and morphogens during syncytial specification. The fluorescent green indicates the presence of bicoid protein that's distributed along the anterior-posterior axis. The figure shows the highest concentration of bicoid at the anterior end on the left side and the lowest concentration on the posterior end on the right side. Depending on the nuclei's position, they are exposed to varying levels of morphogens, and the cell specification depends on this varying morphogenetic exposure. The figure shows both transcription factors, bicoid and caudal, and exposure to the amounts of each morphogen activates one of the transcription factors. Nuclei exposed to higher levels of bicoid have a chance of becoming cells that develop into the head or the thorax, while nuclei exposed to higher levels of caudal have a chance of developing into the abdomen or the tail.